Hi everyone. Can you just put a little comment in the chat if you can hear me all right and see the... Hang on, I've got my iPad going in the background here, sorry. Just put a little comment in the chat if you can hear me all right and see this okay. This is a bit of a... Thank you, Andrea. This is a little bit of a new thing. Last time I did a live stream on YouTube, it was DJ mixes and lockdowns. This is a little bit different. We'll give people a chance to come in. We've got a good number already, which is fantastic. We'll make a start in a few minutes. Thank you, guys. So I might not refer to names in the chat, but again, the chat is there to ask questions. Give me a few minutes and then we'll make a start. Okay, I've unmuted the mic. Hopefully you can hear me now. There's a little bit of an issue there, technical issue, as always. There we go. Sorry about that, everyone. So yeah, I was writing stuff there, but I wasn't actually speaking. Here we go. So yeah, you can hear me now. Good, thank you. So I had a request to begin with the heart. Okay, and that was a request by email. And so I printed off this. You might recognize this diagram as being your classic picture of the heart. And so I thought I would begin here. Now, just to sort of give you some reference points here, the heart is found in B2. So we're looking at B2, which is organization, and the heart is very much part of that topic. So just by way of going over this really quickly, again, some details you have to know about the heart and some key words. So I want to just give you those words that you might want to reference tomorrow. First of all, we say that the heart is a double circulation. Okay, double circulation. And okay, so it seems to be working now. Let me just focus this a bit better. All good. So double circulation is a phrase you want to associate with the heart. And we also want to be able to label the chambers. So a simple thing that we've got to know about is which part's which. And when we look at the human body, we look at this side, even though it's on the left of our paper, we call that the right-hand side. And this obviously being the left. There's a couple of identifying things you have to know about for the heart. First of all, it's the thickness of the wall. And the wall thickness here is thin. The wall thickness in the left side is thicker. And we'll discuss why that is as we go through the chambers. Question, how long the session will be? No, I don't. I don't want it to be too long because you have your own revision to do. It's just a few key points I want to make, Maisie. So it's as long as you want it to be. It's also as short as you want it to be. It is recorded and it does go up in the channel as a live stream. So hopefully, if you want to refer to things, and you can just scan on it and you see what's on the screen as to what's coming up. So let's have a go at labeling the parts of the heart. This is the right-hand side. And the way I think about it is we've got kind of a V shape down here, there, and there. And that helps me remember that these are the ventricles. So we've got the right ventricle down here. Right ventricle, and this is going to be the left ventricle. Now that again, that's how I memorize it. It forms a little V shape down there at the ventricles. And then if those are the ventricles, then these two up here must be the atria, or left and right atrium. So we've got the right atrium and the left atrium. Again, I got these pens tonight. They're reasonably fine, but maybe a little bit too thick for the diagram. Never mind. Now I'm going to use red and blue. The reason being we have to think about the blood. And so we've got the right atrium. Let's begin there. Let's do the journey of blood as it comes in and out of the heart. Okay, so this one here, this is going to be our way into the heart. And that's called the vena cava. And the reason I'm doing it in blue is, again, it shows up reasonably well on screen, is because the blood is deoxygenated. Okay, again, a word that you're going to have to know for tomorrow. Which means there's the red blood cells, there's a bit of oxygen in there, but there's quite a low level of oxygen. It comes in, and then it goes into the right atrium. And from there, it goes into the right ventricle. And from there, it goes out of this blood vessel there, and it goes left or right. And this takes us out to the lungs. And anything that is linked to the lungs has the adjective pulmonary. And because these are vessels going away from the heart, those are arteries. So these are pulmonary arteries. And they go to the right-hand side, the right lung and the left lung. And again, we can discuss what happens in the lungs later on. But the blood picks up oxygen and then it comes back to the heart in the pulmonary veins. I know the diagrams are always a little bit cluttered, but these here are the pulmonary veins. Okay, there's one on the other side, but again, the diagram doesn't show it. Again, it's coming back from the lungs. So we say that these are pulmonary. And I'm doing it in red to illustrate that this is oxygenated blood. So these are veins, okay? Blood travels back to the heart in the pulmonary veins. And again, we'll put in here oxygenated. The blood is rich with oxygen, oxygenated. Okay. And then it goes into the left atrium. From there, it goes into the left ventricle. From there, it gets pumped out this monster blood vessel here. And this goes out to the body. So this is called the aorta. Parts of it go up to the head and the brain. Parts of it go down to the arms. Some of it goes into the core, some down to the legs. So these are the parts of the heart. And these are the blood vessels in and out. We've got 1, 2, 3, 4 vessels. And 1, 2, 3, 4. Sorry, 1, 2, 3, 4 chambers, my mistake. And 1, 2, 3, 4 vessels. Okay, so that's the basics of the heart. And what I would say is you have to know these parts. You have to know the function of them. You have to know the journey, the double circulation of the blood. Oxygenated and deoxygenated. You have to know those terms. And again, the blue and the red is just there to illustrate which way it's going. But let's have a think about why this side is thicker. Okay, why is the left side of the heart thicker than the right? Well, have a look at where it has to go. Because the right side has to pump blood out to the lungs. And so let's put down here lungs. Okay, and the lungs are pretty close to the heart. They're literally sitting right there in front of the heart. Whereas the aorta has to deliver blood to the body. And so we've got to pump blood further distance to the rest of the body, down to the fingertips, down to the toes, up to the brain. That requires a lot higher pressure. So the left-hand side of the heart is thicker because this has to be pumped. The blood has to be pumped at higher pressure. This constricts it. This contracts more. It gives more pressure coming out of the aorta up to the rest of the body. So that's why this side is thicker, because it has to be higher pressure. Okay. Now let's have a think about what happens in the body. Okay, and we can also chat about what happens in the lungs. So this is going right through the paper. So I've got that there. I didn't really think that one through, to be honest. It's gone through 2 bits of paper. That's okay. We can get by that one. Let me get a bit of paper up. And I'll move this out of the way. I've got this background here, the biology book. That should be good. So let's have a think about what happens in the body and also the lungs. Let's have a little chat about the lungs first of all. So we've got these things in the lungs. You get millions of those. Now they're not balloons, they are little air sacs. They're called alveoli. Again, this pen's thickening a little bit. Let's try that again. Alveoli. Okay. Also called air sacs. You might hear them be called air sacs. And in the lungs, you have millions of these. You breathe in air. Air is around 20% oxygen, 80% nitrogen, some bits and pieces of other things. And around these alveoli, these air sacs, you have these blood vessels, which are very thin. And these are called capillaries. And what happens is the lining, the thickness of the walls of the capillary and the thickness of the walls of the alveoli are very thin. And so you've got gas being exchanged across these gaps. So we've got oxygen going from the air sacs into the blood. But the other way, you've got CO2 going from the blood back into the air sacs. Now, it's not one in, one out. Okay, I should stress that. We need to have a think about what's called concentration gradients and diffusion. For example, in the blood, there's still some oxygen, but there's more oxygen in the air sacs. So there's higher concentration of oxygen in the alveoli than there is in the blood. So oxygen will diffuse across this gap. Equally, there's less CO2 in the air sacs in the air that you breathe in, and more or a higher concentration in the blood. So CO2 will naturally diffuse across this gap and into the air sacs. And we breathe out air. Now, we breathe in air, which is about 20% rich of concentration of oxygen. We breathe out about 16% oxygen. We don't use up all that much, but we breathe out certainly more CO2 than we breathe in. Okay, so that happens at the air sacs in the lungs. So this would be the capillaries which branch down from your pulmonary arteries. But also, the same thing happens around cells in the body. So you've got capillaries that go along, and you've got cells that are in your organs or your muscles in the body, and the capillaries run close to those cells, and the same thing happens there. We want to get oxygen into the cells for respiration. We want oxygen in, and we want to get rid of a waste product, which is carbon dioxide. Okay, so let's bring this full circle. Let's have a think about respiration, because the blood's job is to pump blood around. Sorry, the heart's job is to pump blood around the body, and that blood is rich with oxygen to deliver it to cells in the body, the brain, organs, muscles, for our body to do something. Okay, I'm just seeing. Yeah, so guys, if we just keep the chat a little reasonable, I can just kick people off. We're here to revise, remember. Let's just make sure we're doing that. This is very much a trial, and I'm glad people are logging in, but we're here for a reason, guys. Okay, thank you. So we get oxygen. The heart's job is to get oxygen to the rest of the body for respiration. That's the fundamental purpose of the heart. So let's have a think about what respiration is. And this will lie in the B4 topic, the energetics topic. So let's just get another bit of paper. Okay. So what we have to know is a couple of equations, and we're going to have a look at two. They're almost like the mirror image of each other, the opposites. So we've got respiration, and we've got the opposite, or the sort of the mirror image, which is photosynthesis. Okay, in the respiration, we've got two reactants and two products. We've got glucose plus oxygen, and actually we breathe in oxygen in the air into the lungs, into the blood, to that way. We get glucose by eating it, and we'll discuss that at some point. If people want to look at the digestive system, we can do. And these two react together to make water, and I just short to CO2. Okay, so this is what we breathe in through the air. That's what we get rid of as we breathe out. The rest of it, these two chemicals here, we eat glucose, and we get rid of water through sweat and our urine. Now, we say this reaction, it's a bit more chemistry, but we say this is exothermic. Okay, it's exothermic, which means it releases energy. The energy to break these bonds is less than the energy released to make these bonds. Again, it's a little bit of chemistry, which we can do for next week, but this is exothermic. We get energy from this. And this happens in the power cells, the power packs of a cell, which is our mitochondria, which we'll touch upon in shortly. Photosynthesis is the opposite. Okay, we've got water and CO2, and that makes glucose and oxygen. Now, I want to just stress something that's really important when it comes to plants. Okay, and I want to just discuss these two briefly when it comes to plants. Okay, so if we take, let's take a tree. Let's take a tree here. Let's get my green pen out. There's a tree. And if it's really sunny, say it's daylight, so it's a really sunny day, the plant is doing respiration. Hang on, it does photosynthesis. No, hang on a second. It does respiration and photosynthesis. Anything that's alive must be doing respiration. The fact that it's sunny, so let's just sort of do the sun over here. The fact that it's sunny is also going to do photosynthesis. It does both. This keeps it alive, and this gives it the glucose that it needs for respiration. But it makes too much, or it makes more than it needs, so it will store that glucose as starch. Again, we learned about that in key stage 3. That's if it's sunny. If it is nighttime, okay, so let's just get this. Let's do the tree again. Let's put the moon and some stars. Then it's a nighttime, so there is no photosynthesis taking place. It means that the tree or the plant will only do respiration. Is using up that starch supply, using up that glucose to keep itself alive. So it's a bit of it's a misconception that people think that animals do respiration, plants do photosynthesis. No, plants do both. When it's sunny, they do respiration. When it's not, and there's some areas in between. So I want to imagine that there's a sort of a low level of light in between, where it's making exactly the amount of glucose that it needs to do the exact amount of respiration it needs, so it doesn't release any gases out. So here, it's doing more photosynthesis than it needs, so it's going to be giving out oxygen. Okay, so the oxygen levels increase. It's taking in CO2 for photosynthesis. It's giving it more oxygen, whereas here, it's giving out more CO2 because it's doing pure respiration. It's doing this. It's not doing photosynthesis, it's doing respiration. So again, it's just something to bear in mind when you think about plants. It's not just they do photosynthesis and animals do respiration. No, plants do both. Bear that in mind for tomorrow. Now let's have a little chat. Let's see if there's any questions in the chat that people want to know about. I'm more than happy to, if anyone has any questions about any of the topics. There's a few things that I think we could discuss. Let's have a think about, let's see, any questions coming up? Not yet. Let's have a think about the digestive system. Okay, now there are things you have to know about for tomorrow. I'm going to suggest that you have a look in your own revision at the food tests. So how to test different foods for starch, or for protein, or for sugar. There's one for fats, but that's a bit of an odd one. That's sort of for you to do. I want to discuss enzymes. Okay, so let's have a think about enzymes in more detail. And I want to show you, I want to just share my screen with you for a second. I want to share with you, hang on, let me just launch it. So if I go to share my screen, this one here. There we go. So, guys, I apologize for that. I think I figured out when I try and share my screen, the the microphone kicks out, which is annoying. Okay, so let me just try and sort of do a diagram of what I was trying to show you there. Enzymes are very important for digestion. Okay, we eat foods and we take these big, huge molecules into our body, proteins and carbohydrates and fats, and there's no way to get them into our blood. We want to get it into our blood to travel to parts of our body to do the various functions that these foods do. So, how do we break down those big molecules? Well, we use enzymes. So, let's have a think about enzymes in a bit more detail. Okay, it's clear that the animation doesn't actually let me speak over it as well, which is annoying. But it's all right. We can work around that. These are teething problems for another time. So if we imagine, let's have this thing here, and we're going to have there, and we'll have that there. Okay, so this is my enzyme, and let's call this our enzyme. An enzyme is a big protein molecule. Okay, and our body makes them in various places. We can have a chat about where in the body each enzyme is made for digestion. But we'll look at enzymes as a sort of a concept for the moment. And we call, yes, I was just checking something there in the chat. Okay, so we call this part here, we call that the active site. Okay, and we also call, let me just do this, and I'll try and do it in a different color. Let's have something come along like this. So this is my food molecule, and this is called the substrate. Okay, that's the name of it. It's called the substrate. And this is going to come along, and that's going to fit in exactly into this gap in the enzyme. And the enzyme will, because it's locked in place, it can split it apart. Okay, so this is the enzyme with the active site. This is the substrate, which is the molecule we want to split up. This comes in here, and then it splits up into two separate smaller molecules. So it comes in, and then it goes off into two. And that's the enzyme. That's simple as that. The key to it is these enzymes are specific to these substrates. These molecules fit exactly into here. If I come along with another molecule, if I come along with one like this, then this molecule, this substrate, will not fit in to this enzyme. Okay, this one did not get the right shape to fit into this enzyme. So this cannot be broken up with this enzyme. This one can, this one cannot. So that's a little basic thing about enzymes. But let's think about the words denature. Okay, so denature, that will come up tomorrow at some point. Denature, what does that mean? Well, in terms of this enzyme here, denature means the shape will change. So we could have something like this. It goes from this shape to this shape. And it means that the enzyme can no longer do the job that it was intended for. It can no longer have this substrate fit into it to break up. Okay, so this, we call this denatured. The shape has changed. Okay, the shape alters or changes, and it cannot do its function anymore. But what could cause that? Okay, there's a couple of things that could cause that. It could be that it is too high temperature. Okay, temperature's too high. That's one reason. Another reason could be the pH is too low. So that would be acidic, or too high. That's going to be alkali. If the environment for your enzyme to do its job, if it goes too acidic or too alkaline, then it won't do its job properly. It denatures. Too high temperature changes the shape of the protein. It doesn't do its job anymore. It denatures it. There's one last one, which is the if it goes too cold. Too cold means that these things won't be going fast enough. Okay, so they're going too slowly to lock in place and break apart. Whereas if it's getting warmer, they'll go faster, they'll lock in place and split apart. But if we make it too high a temperature, denature the shape, or the enzyme is denatured, and if it's too acidic or too alkaline, also denatured. That will probably come up tomorrow at some point, that kind of question about denaturing of the cell. Okay, so guys, that's about half an hour so far. If there are any questions, please let me know. Again, if the chat could be reserved for people that want to ask questions, that would be great. I don't want to chuck people out because that's not what we're here for. Okay, this should be for help and revise. But I see people are still treating the class seriously as they do in school, so let's try and do a little bit more work then, folks. Okay. Thank you, Nicola. Yes, denature. Now, that's a couple of sort of topics done. Okay, so question where enzymes are released from. Excellent. That's a good question. That's the kind of thing we need to do to find out where they come from. So in the digestive system, we start at the mouth and we end up, well, we end up down here. Yeah, we end up, I don't want YouTube to kick me off, so we know where we end up. But along the way, certain things happen. Straight away in the mouth, we have saliva. And saliva is going to make an enzyme, which is called amylase. Okay, an amylase is a carbohydrate. And as the name suggests, carbohydrates is the function of it is to help break up carbohydrates. Okay, a cool little test could be if you put a bit of bread in your mouth, which is the carbohydrates, then the amylase will go to work. It will help to break up the starch that's in the bread, the carbohydrates, which is starch. It will break up that into sugars, and the bread will eventually taste sweet just sitting on your tongue. Okay, it's a little test you could try. Again, it's not going to be essential for the exam tomorrow, but it's good to know that the saliva made in the saliva glands makes amylase, which is a carbohydrate, and that is to break down starch. Okay, so that is to break down starch. That's a specific example of carbohydrates. Along the way, we've got other organs. We've got the stomach, and in the stomach, we've got, let's do over here, we've got a protease. And protease is to break down proteins. Okay. And proteins, so I should just say actually over here, let's just go back up here for a minute. The starch is broken up into sugars. Okay, because I think it's important to know what it breaks it up into. Protease is going to do the following. Proteins are broken up into amino acids. These are the building blocks for other proteins. And also, along the way, you've got this thing here called the pancreas. And that is a powerhouse. Sorry, I was focusing on something else there. There's no E at the end. Please don't get it wrong. The pancreas makes all the proteins and all the enzymes. So we've got our amylase, we've got our protease, we've got the lipase as well. So we need to mention that. And lipase is made in the liver. So let me just check this. Okay, so again, I'm referring to some books as well because there's a lot to remember, and you've got to always revise stuff. You're always checking things. So lipase, the enzyme is the liver and the gallbladder combined. So we've got Bile is produced by the liver and stored in the gallbladder before being released into the small intestine. It's not so much an enzyme, but it does help break down large fat sort of globules into tiny droplets. It's called an emulsifier. It's called emulsification. And the bile also neutralizes the acid, because this here is going to be a low pH. So it goes acidic in the stomach. You want to neutralize that. You want the digested food to go through your system at pH neutral after the stomach. So again, I would look up these in more detail. So you're looking at your digestive enzymes. Okay, there's a lot to get into. I want you to revise that, but it's basically you've got protease in the stomach, amylase in the mouth. The pancreas makes all of them for the rest of the intestine. Now, I want to focus on B3, which is infection and response. I want to do a little bit of work on that, just to help you out when it comes to those things. So let's grab another bit of paper. Put some here. So my recommendation, folks, is I want you to look up the digestive system yourself in your revision, but bearing in mind enzymes and denaturing, that's going to be a critical part of that whole thing. That's sort of the hardest part there. Yeah, so the chat is getting a little bit distracting. Again, people aren't using it for what it's for. This is a shame. All right. But these things do happen, unfortunately. So for those that are paying attention and want some help, please ask any more questions. That's what we need to do. But I want to have a look at infection and response, because this is a huge thing. Infection. I think that'd be better if I spell it right. Let's try that again. Again, the chat is quite distracting. Okay, so infection and response. And we're looking here at what's called communicable disease. Okay, communicable disease. And just a question there. What are trophic levels and why important? Are trophic levels in B4? I don't think they are in. I think that comes in the paper 2 when we look at ecology. Trophic levels are in food chains. So you have the producer, then you have the primary consumer, and then secondary consumers. As you go up the trophic levels, the energy efficiency is quite low. So that's why you have to eat a lot of the previous trophic level. Tomorrow will be about cells and cell transport. So what a couple of things to do, folks, in the time remaining, which I think is going to come up tomorrow. We're going to do a little bit on communicable disease, and then I want to do a little bit on cells, but in particular, what's called cell transport, things that travel in and out of cells. There's a big required practical called osmosis, and that will come up at some point. So I want to cover that. So 2 major things to go, and then we will have a look. Again, guys, the chat isn't particularly helpful. We're doing chemistry before the chemistry exam. This is biology. So again, feel free. I can kind of see in the chat the genuine questions, but there we go. We can't please everyone. So communicable disease. Now, a phrase we could use for this one would be, you can catch it. Okay, I wouldn't use that phrase in the exam tomorrow. Certainly, communicable disease is what you want to look at, but it are things that you can catch. You can catch it from other people. The main way in which that happens is through these things. Pathogens. And a pathogen is a microorganism in which can cause disease. Okay, patho implies death. Gen is the sort of the thing. So these are microorganisms that can cause death, and there are 4 that you have to know about. Okay, I'm not going to make this into a test or a class, but the 4 are: we've got bacteria, we've got virus or viruses, we've got fungi, and we have protists. Now, your job is to have at least 2 examples for these. That's a trickier one because we tend to only know malaria for this one. But you have to know about pathogens, and you have to know some examples. Okay, so you have to know some examples for these. My go-to one, now what's interesting is when this GCSE first came into being, this was not a thing. Okay, but it's a valid answer now. COVID. In terms of bacteria infection, we could have salmonella, which is food poisoning. And we could also have this one here. Let me get my spelling correct. It's a trickier one to spell. Yeah, this is OEA. That's an STD. And salmonella is food poisoning. COVID is a virus. We could also have, there's many other viruses that we could think of. We've got this one here. HIV, which is called human immunodeficiency virus, that can lead to AIDS, which is acquired immune deficiency syndrome, where your immune system is depleted and you could catch something as insignificant as a cold and die from that. COVID, HIV, we could have measles and the viruses. Fungi, you've got things like rose, black spot, and that is purple or black spots on leaves of plants, and usually it's a fungal infection. The leaves drop off, photosynthesis decreases. There's also, you could have this one here, which is athlete's foot. That tends to be sort of itchy skin. That's between the skin of your toes. If you have your feet in your socks all day, or you know, I'm not saying you share socks, that's weird, but the environment, that hot, humid, damp conditions, that's perfect for spores of fungus to reproduce. So that's fungal infection. Protists, we need to know about malaria. Malaria is caused by the Plasmodium protist, which is kind of a parasite. It goes through a life cycle in the human body. Mosquitoes, it's only really, I think it's the females of one particular species that can bite humans and get some of the infected blood, but it's a parasite and it can cause extreme fever and death in humans. It is reckoned that all the humans that have ever lived, half the humans that have ever lived and died as a result of malaria. So mosquitoes are responsible for half the human deaths has ever been. That seems quite a sensational number, and there's nothing to say otherwise. But that's how grim malaria is. It kills a lot of people every year. So my dogs just come in the room having a nosy at what we're doing. So these are the pathogens, but let's think about what's called transmission methods. So how do these things actually transmit? We'll do it over here. So we've got airborne, but you need to be a bit more specific than that. It's not just in the air, but it's in water droplets in the air. Okay, it's in water droplets. And so that is the main delivery method of viruses. Okay, we could also have, so that's airborne. We could have contaminated food. We could have contaminated water. In the event of a natural disaster, so say there's a devastation somewhere around the world, one of the most important things to establish is a healthy and clean water supply. Otherwise, things like cholera could rise up and infect the water, and could that sort of secondary disease that can harm a lot of people in the area. So there's contaminated food and water. Airborne is water droplets. There's contact. direct contact, and that also covers sexually transmitted disease. And there's one last one, which is a vector. And a vector is another organism which transfers the pathogen. So, example, in mosquito, thinking it back to malaria. So, the vector for malaria is the mosquito. Now, the mosquito actually digests the protist, the parasite, in its stomach. So it doesn't actually affect the mosquito; it affects humans. So as a mosquito bites human and human, it passes on the parasite without actually harming itself. So a vector is an organism which can pass on the pathogen from one person to the next. Okay, so that's a few things on how these pathogens are transmitted. Let's have a little chat about defense. Okay, because this again, I've got a funny feeling this is going to come up tomorrow. So let's have a chat about the lines of defense. Okay, so our first line of defense is our skin. Okay, so we've kind of got these layers. Let's do it from this way. So we've got pathogens. Let's just do them over here. There's my pathogen. There's a virus and a bacteria. They're not to scale because these would be much bigger than these. But the first line of defense is skin. Okay, so skin it's also got antimicrobial secretions, but the skin is a barrier. All right, so skin is a barrier. We've also got our tears. Okay, so we could have also our tears, and our tears contain an sort of microbial enzyme. I can't remember the name of it. I don't think I have to know the name of it, but our tears also help fight infection. If you think about your eyes being really exposed to the environment, our tears, which again, your eyes are always producing tears, have these antimicrobial secretions. So those are tears. If the pathogens get past the skin, they're going to go in through our nose and our mouth. And so we have another layer, or next one down, which is, let's just see how we're going to write this one. We've got, if I can try and do it like this. So we've got, so this is an airway. We've got cilia cells. And these are so in the airways in our nose and our throat, and we've also got mucus. So we've got mucus. Mucus is the sticky snot, basically, and the mucus can trap. So I'm just having a check in the chat here. Okay, guys. Again, we're here to try and learn something, pick up some ideas for tomorrow. The mucus in your nose and your throat will trap any microbes, so any bacteria, viruses are supposed to stick to that, and the cilia cells will sweep the hairs, will sweep up the mucus up your throat, whereby we swallow it into our stomach acid. And a stomach acid has a low pH, which will kill almost all. I can't say everyone. It's 99.9% effective. It'll kill almost all microorganisms. So our mucus in our nose and our throat catches it, sticky. Our cilia hairs and our lines our throat and our nose sweep the mucus up to our throat. We swallow it into stomach, and then that will kill the pathogens. Now, that's the sort of first line of defense. That's the skin and the ways into the body. The second line of defense is our immune system. Okay, so if it gets past this line of defense, then we have to rely on our white blood cells. Okay, so that's the first line. Second line is down here. Now, white blood cells, there are three sort of main functions of white blood cells. Okay, one, they will get infected. So they can get infected deliberately. They can get infected deliberately and then self-destruct. They can engulf the pathogen. So they can self-infect. They can get infected and then self-destruct. They can engulf the pathogen. They basically wrap around the pathogen. So if I can try and do a little diagram over here to the side, I want to imagine this is my pathogen. Here's my white blood cell. It'll come along and it will start to wrap itself around it. So that's going to be 1, that's going to be 2. Eventually, it's fully enveloped around it. So it'll come along, it'll see it. These things actually hunt them down, actually chase after them. It's quite cool. There's a few YouTube videos where you see white blood cells chasing bacteria around the blood. It'll catch up with it, it'll wrap around it and engulf it, and then it will basically destroy it from wrapping around it. So that's one job. That's engulf pathogens and destroy. And the third one, which we're going to spend a bit of time talking about, is it produces antibodies and antitoxins. And I want to spend a bit of time looking at this one here. Okay, so white blood cells can make antibodies. Now let's have a chat about what those actually do, because these help fight off particularly viruses by helping number 2 to happen. So how can we catch a virus that is too small and too quick? Because viruses basically they invade a cell, they hijack the cell's machinery, the genetic machinery, and they copy themselves thousands of times, and they kill the cell, and all the viruses are released out of the cell into the body, which can then go on to attack other cells. So viruses are fast reproducing. They're not living things. They are basically protein sacs with DNA in it. So they're fast reproducing pathogens. Okay, let me just have a look at the chat here. Again, I'm not going to block anyone. Again, I'm trying to sort of focus on this as well. It'd be great if you can keep the chat for what it's here for, which is asking questions. I'm trying to go over this, but let's have a little chat about antibodies. I need another page because this is pretty cool. So let me go to this page here. I'll come back to this one in a minute. So if we think about a virus is not a living thing. A virus is a protein sac that has some DNA inside it, or RNA, or some genetic material. Okay, so this is genetic material. But the thing about a virus is they will have these spikes. Okay, I can hear my dog barking outside. I do apologize if you can hear it. So this is a virus. And these are far too small and far too fast to actually catch. So we need to find a way to slow these down to make them easier to catch if you're a white blood cell. And the way we do that is one white blood cell is going to help make things called antibodies. I should just say that these spikes are called antigens. And they stick out. Okay, they are protein spikes that stick out from the virus. But our white blood cells, one particular type, is going to make antibodies. And antibodies come along and they can attach onto these spikes. And these are called antibodies. Now, these antibodies are unique to these antigens. That's important. So the antibodies are unique to antigens. There is a spike for COVID. There is an antibody for COVID. There's a spike for the flu virus. There's an antibody for the flu virus. Actually, the annoying thing is the flu virus is always mutating. These spikes are always changing shape. So we always have to make new antibodies. But here's the thing. If this happens naturally in the body, then antibodies are made at a rate of 2,000 per cell per second, and they stick to the antigens. Now, these are sticky at both ends. They stick at this end, they also stick at this end. So, what to imagine, we've got a bunch of viruses. There's my viruses over here, and they have all got attached to them antibodies. But along comes another virus, which itself has its antigens. It's the same type of virus, and you have these antibodies stuck to it. These antibodies stick to each other. And so what happens is you've got dozens, hundreds of virus cells surrounded by antibodies, and the antibodies stick to each other. And so this is not just one single virus, this is a large ball of viruses. And this becomes larger. Because it becomes larger, it also becomes slower. Now, if it's slower and larger, it becomes much easier for the white blood cells, this one, to come along and engulf and destroy. So, in summary, our bodies make antibodies. Our white blood cells make antibodies which stick to the spikes on a virus. The antibodies stick to each other, forming little clumps of viruses which become larger in size and slower in speed, much easier for our other white blood cells to chase and to engulf and to destroy them. So that's the purpose of antibodies if it's done naturally. What about if we don't recognize the spikes? Okay, what if our bodies can't make an antibody that matches up with these spikes? And there we have a problem. Because how do we learn how to do it? Well, there are two options. And let me discuss those two options with you just now. And I appreciate time is moving on, guys. So I'll do this bit, and then we'll do a little bit on cell transport, and then I'll leave you to do some of your own revision. But let's think about, what if we don't know the antibody that we need? Along comes a virus and we don't recognize it. We have two options. One, time. Okay, here's the virus, and there, here's the spikes. We could wait. Okay, we could wait for our bodies to make the antibodies. Now, the danger there is if we wait too long, we could get really sick. Okay, and the time it takes us to make the antibodies, we could fall more sick. We could actually, again, in some cases, let's not beat around the bush, we could die from it as well. So the danger there is time. If we just say, let people get ill, and they will naturally make their own antibodies. I'm just going to just block a few people here. Okay, block person. So that's one option, not the best option. The second option is vaccine. Now, my job is to present to you what a vaccine is. We're not going to get into discussions about what people feel about vaccines. This is just about what a vaccine is. So let me explain it. A vaccine is when there's various types. Again, the COVID ones are a bit more complicated, but let's go with a really basic version of a vaccine. It is a dead or inactive virus. In other words, it could be just the cell, or the shell, I should say, and the spikes. And this is dead. It's empty. There's nothing in there that's going to hijack a cell or going to make the person sick. But what it does do is the body says, okay, there's something new. I don't know what it is, but I'm going to learn what I have to make to attach antibodies to it. So in other words, the body learns the type of antibodies that are needed for this particular virus. And so that in the future, along comes the actual real virus, which has the genetic codes, and the body goes, aha, okay, I know what to make here because I've learned to recognize these spikes. So that if I do get the actual disease, I know that I have to make these antibodies. I should have used my different color. I know to use these to make these antibodies for this virus should I get it again. And if I do get it again, which I've got here, I know exactly what I need to make. Okay, so that is the intention of a vaccine. We get a dead or inactive version of the virus so that our body can learn what it needs to make in terms of antibodies to attach to these spikes, these antigens on the virus. This is not a real virus. It won't duplicate, it won't cause any harm. It might feel a little bit funny because the body does do what's called an immune response. So it might feel a little bit feverish because the body's saying, okay, this is new. I want to learn to make antibodies. But along in the future comes a real version of the virus. And because we've learned what we have to make, we make the antibodies. And it can help us fight off this infection if we do get it. So that's the purpose of a vaccine. There's a few variations. For example, the COVID vaccination, there's one version which was quite smart actually. Our bodies were injected with the genetic code that only made the spikes. And so we basically mimicked our own cells were pretending to be COVID viruses. So we made tons of spikes. So we actually taught ourselves what antibodies we needed for COVID. That was a really ingenious way of doing it. Again, I'm not going to get involved in the debate of it. I'm just presenting the facts. It's up to you what you do with information. Now, I think we'll do one last thing. Okay, I can see people getting a bit tired in the chats, and people are using it for social functions. Again, that says more about you than me. Okay, I'm trying to provide something to help you out, guys. If you want to have a social chat, then by all means, that's your call. But please do it in private, not on a revision chat. So let's do one last thing. And then if you have any more questions, I'm happy to go over it. So we're going to look now at what's called cell transport. Because as I mentioned way back at the start, we've got to get things in and out of cells. We're going to get gases in for respiration. We've got to get glucose in for respiration. We're going to get gases out for waste products. We're going to get water out. So we need to think about how we get things in and out of cells. Now, let's present three different things. Firstly, we're going to have what's called diffusion. Diffusion. And the way I'm going to present this is as follows. We're going to have a membrane. This is my membrane of my cell. I'm being very careful about my language here because I want to use a different word shortly, but this is a membrane. And let's say we've got some red molecules. I don't know what they are. There's just a bunch of molecules here. And we've got lots and lots and lots on this side. Over here, we've got not as many. And so we could say, in terms of concentration, this is high concentration. This would be low concentration. And what happens is these molecules move across both ways. That's important. They go both ways. These molecules go across to this side. These ones go across to this side. But where have I got more of them? I've got more on this side than over here. So on average, there will be a greater movement from this side across to here. Okay. So we go from high concentration to low concentration, and that's called diffusion. Okay, so think back to way back at the start. I've got the sheet here actually. Go back to this one. I like to imagine this could be oxygen in the air sac, which is oxygen in the air sac. This could be oxygen in the blood, and we have an overall movement of oxygen from the air sac into the blood because there's higher concentration in the air sac than there is in the blood. Air sac and blood, with oxygen moving across. Okay, and this, we can call it passive. Okay, it happens naturally because of concentration. I like to imagine it as, again, going back to my physics hat on, if we can think about a ramp, a ball will naturally roll down the ramp from high to low. That's just how things work in physics. They go from high to low. Okay, let it go, and it will naturally go down the slope. That's called diffusion. Let's do another one. This one's called active transport. Again, we have a membrane. And let's choose green for this one. Let's have a few over here. Let's have many more over here. Okay, so clearly we have high concentration on this side, low concentration on this side. But what happens here is this moves across to here. Now that seems to be the opposite of diffusion, and it is. The way I think about it is I want you to imagine that we have a ball going up the ramp from low to high. And the way I like this little visual reference is because that requires energy. Okay, this requires energy. You have to physically push the ball up the ramp. You have to use energy to push these particles across where they want to go this way on average. They want to go from high to low, but we're pushing them against where they want to go. And so this requires energy. And this could be nutrients in the soil, where you have more of them in the plant than in the soil. You want to push more into the plant. That requires energy to push them against where they want to actually go. So they want to go this way and diffuse, but we're pushing them against that, what's called the concentration gradient. Okay, there's another phrase you might want to include tomorrow. The concentration gradient. Okay, the gradient is diffusion goes from high to low, which is down the concentration gradient. Active transport goes up the concentration gradient from low to high, but that requires energy. That's the key inclusion for active transport. Now, there's one more. These involve molecules in and out of cells, and you can do that through little things called channels. But let's do one last one. Actually, the third one. Now, I'm sure people are ahead of the game in this one, and this one is called osmosis. Okay, osmosis. And that involves water. Now, I want to just give you a few key phrases that you've got to include. Now, I'm going to draw this as a little dotted line. And this is called a semi-permeable membrane. Because our cell membrane actually has little gaps in it. Now, they're really small, and they allow things like water or oxygen or CO2. So these things can get through, but things like glucose or other molecules, we could have salt, these things do not. So the gaps in the semi-permeable membrane allow the small things to get through, not the big things. Okay, so let's have a think about what happens in osmosis. And I want to think about salt. Okay, I want to imagine this is water. And this is also water. And that's a fair assumption. Our cells are full of water. So our cells are full of water, but we have things dissolved in it. So I want to just I'll do this in pink actually. Let's do it in pink. And let's do a lot of these molecules over here. And let's go for a few over here. Now, what's happening is we'd say that there's a high level of dissolved molecules in this water. So we say that as hypertonic. So hyper means a lot or high. Tonic just means things dissolved in it. Again, I'm being a physicist. This means there's a lot of things dissolved in the water. This would be hypotonic, which would mean low numbers, a low amount of things dissolved in the water. And what happens is water will go from this side to this side. Water goes from where it is almost quite pure, so not a lot of dissolved things in it, to where there is a lot of dissolved things in it. So water will go from here to here. Okay. Now, if we think about this as not just a diagram, but what about an actual cell? So if we think about these cells here, okay, these are our red blood cells. Now, I want to imagine that we have quite a lot of salts or even quite a lot of sugar in our blood. So this could be our red blood cell, and this could be our blood, in which case water will leave our red blood cells to enter the blood because there's less dissolved things in our cells than there is in our blood. We get lots of sugar and salt in our blood. So what happens is water will leave this cell to go to this one, and that means that red blood cell will shrivel up and not do its job properly. It won't be able to hold oxygen. It will shrivel up and die. Alternatively, what if we have really sort of a lot of water in our blood? We've drunk a lot of water, probably more than we should have, and this becomes our blood. We get very few things dissolved in it, and then these could be our cells, in which case water will leave the blood to enter our cells, and that will cause these things to swell up and they could burst. Again, the end would be you can't use the cells. So you have to be careful. You have to be careful with your blood glucose levels. You do that in paper 2. You have to be careful with your salt. You have to be careful with your hydration. Okay, hydration is good, but way too much water can be bad. Equally, we need salt and sugar in our body, but too much of it can also be dangerous. So osmosis, now the osmosis required practical is not using red blood cells, but just using either. Now, actually, this is a bit of a controversy a few years ago, where we kind of teach it with potato cells. And so we have chips of potato, basically cut them into blocks, and we place them into beakers of salt solution. So we could have 0%, 100%, and 50% salt solution. And by placing the chips into these beakers, so in other words, you place the chip into there, then there's a certain concentration of stuff dissolved in the cells of a potato. This could be a potato chip. So the cells have a certain tonic level, a certain amount of chemicals or molecules dissolved in its cell water in the cytoplasm. When you put it into pure water, then I want to imagine this is the potato and this is the water outside. Water will enter the potato, and so the potato gets larger in size. Whereas over here, when it's really salty water, this could be the potato, and this would be the salt water. Water would leave the potato to go into the surrounding water, and that will make the potato smaller in size. And so we usually do this with 3, 4, 5 maybe different concentrations, and we measure the percentage change. Now, just a couple more things before we hang up. Percentage change is a really important thing, and not just in biology, but also in chemistry and physics. And it is the final value, take away initial value, divided by initial value. And that's going to be times 100. So final, take away initial, divided by initial. So let's just say, for example, this was 50 grams, and they put it in that, and it went up to 60. Okay, it'd be 60 take away 50 divided by 50. Okay, which is 10 divided by 50, which is, even I can do that, it's going to be 20% increase. Whereas over here, let's say it went from 50 down to 40. That would be 40 take away 50, which is negative 10, divided by 50, which is going to be negative 20%. So it decreased by 20% from its original value. And so, again, this is maybe for the higher work, you're going to get a graph like this. And this is going to be our concentration, and this will be our mass change in percentage. You're going to get something like this. And so you have if you chart all these concentrations, say we did 5 beakers, then you would have some kind of a line going down like this. So this might be your pure water, then it's going to be your salty water. This is the different ones in between. And we can actually find where it crosses the line. That's going to be the same as the potato concentration. That would be the same concentration as it was in the potato. So you found that exact, it's called isotonic. So isotonic value. Iso means the same. Tonic means dissolved chemicals or salts dissolved. Isotonic value is when you have exactly matched up the potatoes concentration to a value. Now I went a little bit quicker there, but again, please do your own revision. I'm going to leave it there, folks. I can see people are still chatting away there. Now, the test is tomorrow, and I've gone over a few of the big points. So your job then is to do your own revision. Again, guys, I'm trying this out tonight. Okay, my plan is to do this for chemistry and for physics. Being a physics teacher, this is my least competent subject. Chemistry, yep. So chemistry is going to be paper 1, which is bonds, the periodic table, and quantitative chemistry, as well as electrolysis and bond energies. That's easy. Again, easy. I shouldn't say easy. It's easy for me to do a revision session. This session tonight is a lot of words. Remember, biology is not anything other than the language of describing things. Okay, we're looking at a language tonight, and there's a lot of words. So my recommendation tonight, folks, is as follows. Read over your notes, read over any revision guides, try and learn the keywords. I've given you a lot of keywords tonight, just by way of a little summary. In fact, you can go back over the video, just skim over the video, because it will come up shortly onto the YouTube channel. I've written a lot of keywords down there. I've tried to show you it in the right context. Your job is to do some revision tonight. If you're at Mayfield, then we'll get a few booster sessions before lunch. If you're at Admiral, I'm sure you have equally similar things to do there. So good luck tomorrow, folks. Remember, it's 1 paper out of 6. Your total score is out of 420, and you'll get a grade based on your total score. So try and do your best in every paper. But this one tomorrow should be good. I have every confidence in you, and I'll leave it there, folks. And if you have any questions, please ask your teachers tomorrow in your own schools, or there's a few things on YouTube, for example. There's cognito-science. There's obviously the GCSE revision videos. I like cognito-science. Question there, what mark out of 420 for different grades? That information changes every year, Thomas. So, for example, the numbers based on everyone that sits the exam. So, basically, all the scores. But let me just, okay, I'll just divulge the information to you since you've asked. Let me find a bit of paper. So, if we think about all the grades, so we've got, let's see, we've got numbers. Hang on, we get grades down here. And we've got numbers of students. And what happens is, so these are the grades. This is going to be your 1 to 9. And combined case, it'll be 1 to 9 9. And we tend to find something like this. And so there are lots of people in the middle, fewer people down this end that getting low grades, fewer people up here getting high grades. And the government say, right, what do we need to satisfy a 4? And also, what do we need to satisfy a 5? And what do we need to satisfy a 7? And so, out of 420, the exam board will set a number that you have to get out of 420 to get a 7. And that also means 6s and 8s and 9s and 3s and 2s and 1s. It'll also say, what do you need to do to get a 4? Now, I don't want to say, get this number and you'll be sorted. What I would say is, get as many marks correct as possible. Don't think about accumulating so many scores out of 420. Try and just think, what can I do to get the most out of this paper? Okay. So, to get a grade 4, again, I can't remember off the top of my head. It's not as high as you think, but I don't want to say that to people and people go, okay, I can do a little bit and get a good grade. Try and get as many marks correct as possible. Okay, try and get as many correct as possible. I know to get a grade 9, it's up around the average of about 60 marks per paper, if not a little bit lower. But I'm not going to commit to anything just now, guys, because it does change. It depends on everyone that sits this year. So this number here might change. It might be further up, it might be further down. We just don't know. So don't bank on getting a score and getting a grade, because it can change. All right. But that's all secondary to trying to get the highest possible number of marks. If you focus on getting the best number of marks, then that's the right attitude to have. Okay. So, guys, thank you very much for your participation. I'll have to have a think about using the chat function. I want to use the chat function, but obviously some people can't self-regulate. Yeah, just like to point out, guys, that you are year 11s, you have an exam tomorrow, and so hopefully most of you are going to take this seriously, which I'm sure you will. But good luck tomorrow. Ask your teacher any questions that you get overnight in your revision, and I will see you on the other side. We'll do this again before chemistry. Okay, thank you, guys. Hopefully it was helpful. Again, it's a little bit on the spot. These things, we're trying to do some new things here. Yeah, so good luck, folks, and I will see you on the other side.